All right, so this is where we left off last time. We were talking about the innate immune system, and that means it's the I don't know what's happening. Yeah, that's better. So one of the things that your innate immune system can do is initiate an inflammation response. So have you ever experienced inflammation? Give me an example. Something that happened to you and then you had inflammation. Say again?
Surgery.
After surgery? Did you get like a kind of a redness or, yeah, so at the site that was cut, that's pretty common, right? Anyone else? Yeah, and you scratch it and then it gets all red. Yeah, so any break in the skin is the most typical way that you get an inflammation response. And what will happen is there's some sort of puncture, then bacteria and other microbes get into the cut or the abrasion, and that sends, because the tissue is actually damaged and because there are foreign things that in your body that shouldn't be there, below the surface of the skin, below the place where it's normal to have microbes, that initiates this response. That includes the following things. So one thing that happens is a lot of white blood cells come to the area. Some of them are mature, some of them are not, and as they arrive, they mature. They're stimulated to mature by what's happening. There's also clotting factors, which is why you don't bleed to death every time you get a paper cut. And this is not just to prevent you from bleeding out, but also the clotting prevents microbes from getting any further into your body. Now, we've all had this experience, right, where in those sort of later stages of an inflammation, especially if it's scabbing over, there can be like pus and a kind of fluid in the area, swelling, and that's because dead white blood cells and dead tissue are accumulating. This is just a bunch of dead matter. We can have an acute inflammation response, which is like, you get a mosquito bite or you get a paper cut and then it sort of rapidly inflames and then it heals. But you can also have chronic inflammation, which isn't so good for you usually. So what are the things that are happening in an inflammation response? There are a variety of proteins called acute phase proteins that are activated. And it's important to understand that these proteins have these very specific roles, but they're usually either not deployed or they're in your body but not activated, because we don't want to have inflammation all over your body all the time. And we know that there are chronic conditions that are really damaging to your body if this is going wrong and it's overactive. So we're going to talk about the complement. We're going to also briefly talk about cytokines and kinins. But most of our effort or our time will be spent on understanding the complement. Another thing that happens is vasodilation. What does that mean? Vasodilation. Yeah, blood vessels are basically expanding. Why would that be helpful? What's the point of that? Yeah, we want more blood flow. Specifically, we want more white blood cells to come. Vasodilation also tends to increase the permeability of the blood vessels so that it's easier for the white blood cells that are traveling to be grabbed onto or grab onto the inside of the blood vessel and then squeeze out into the tissue, right? So they're circulating through your body, and in these areas where the blood vessels are dilating and are more permeable, they'll find an easier time getting into your tissues. This is triggered by a variety of chemicals that are released by cells. So histamine, kinins, prostaglandins, and leukotrienes are all chemicals released by cells that can trigger your blood vessels to dilate. We know the effects of inflammation. We've all experienced them. The site gets red, there's swelling because there's lots of fluid come to the area because we have expanded blood vessels. It's also painful because pain receptors in our skin are being activated and it's releasing heat. So there's a couple of different cartoons that are included in this PowerPoint presentation that you can look at, and they sort of cover the same series of events, but basically you have a break in the skin, you have microbes coming in with that break where they shouldn't be. That tissue damage that's a result of that is causing the release of some of these chemicals that I just mentioned, histamine, kinins, prostaglandins, leukotrienes, and cytokines, that are then signals. They're signals that are sent out and they call white blood cells to come to the site. So it's like the 911 call and the first responders all come. We have clotting factors in the blood that are forming blood clots in the area. In the later process of healing, we get an abscess, which is our body's attempt to initiate the healing process. And of course, as I just mentioned, one of the things that happens when you release all these chemicals is that your blood vessels dilate and become more permeable. So these are these chemicals I just mentioned: histamines, kinins, prostaglandins, and leukotrienes. This is a summary of what they do. So they have slightly different effects. So some of them cause vasodilation directly and increase the permeability of blood vessels. Some of them help histamines and kinins to do what they do. Some of them actually increase the ability of white blood cells to attach to things. But all of them essentially are part of the process of bringing white blood cells to the site. Now, once you have white blood cells rolling through your blood vessels, how are they actually getting into the tissue? And I talked about this last time. So they, when they receive these signals, there's a communication between the cells here that line the blood vessel wall and these white blood cells. They grab onto each other. These guys get kind of flat and they squeeze in between the blood vessel cells to get out into the tissue. And they're responding to chemical signals that are coming out that are sort of saying, hey, this is where the problem is, please come and help out. And what are these things that are coming? Well, some of them are macrophages that are going to engulf microbes in the area, so get rid of that bacteria or those viruses or whatever that are coming in. And some of them are lymphocytes that will be important for establishing a more specific immune response, which we'll talk about in chapter 17. Some of them, as I mentioned, are immature. So this monocyte is an immature version of what it's going to be. Once it gets to the site, it matures and becomes another kind of phagocyte. And this is, again, something we're very familiar with, part of the later processes of tissue repair, repairing that skin. We have a scab. We have a blood clot underneath the scab. We have the skin cells starting to regenerate and to sort of bring everything back to normal. So that's inflammation. Now there's also another part of your innate immune system, which is fever. And sometimes people get inflammation and fever mixed up because fever is also associated with increased temperature. But inflammation is a local response to a cut in your skin, usually, whereas fever is a systemic, basically, increase in body temperature. Does anybody know what's actually responsible for setting your body temperature? What's your thermostat in your body? Yes, it's the hypothalamus. It's this tiny little structure in your brain. And if you can, usually it's set to 37, right, for most people, 37 degrees Celsius. And if you can actually reset it and shoot the temperature up. So it's normally at 37. How do you reset that temperature? A common way to do that is a gram-negative bacterial infection. So this endotoxin in gram-negatives, remember, is where? Where is that gram-negative endotoxin? It's in that outer layer of the cell wall, right? Remember that gram-negatives have a normal plasma membrane, they have a thin layer of peptidoglycan, they have that second phospholipid bilayer, and on the outside of that second layer is the lipopolysaccharide layer, which is basically just lipids plus some sugars. And one of those lipids, lipid A, is an endotoxin, which is toxic when it's released when the cell's dying. So that endotoxin, when gram-negatives are actually being killed, the endotoxin is what causes the phagocyte that's grabbed onto it, engulfed it, chopped it up. Then that endotoxin causes this phagocyte to release a chemical called interleukin-1. And that interleukin-1 goes to the brain and talks to the hypothalamus. Because it's received this signal, the hypothalamus releases this other chemical, prostaglandins. That's not the only time you release prostaglandins. We talked about tissues in the body at the site of inflammation releasing prostaglandins as well, right? So it's having different roles in different parts of the body. But the hypothalamus releases prostaglandins and resets itself to a new high temperature. Now, what's interesting is that what happens as a result of that is, first of all, the rate of metabolism goes up. Why do you think that would be the case? If we add a little heat, why is metabolism increasing? Yeah, the enzymes work better at a higher temperature. Now, we can't go too high, otherwise what happens? all of those enzymes will fall apart. So it's a really tight window, right? We're going to boost the metabolism a little bit, but we don't want to go too high because then everything's going to die. And one of the things that happens is that we start shivering. And this is, you may or may not know this, coming from a very cold climate, I actually know this, shivering will make you warmer, right? So you can shiver even though you're not cold. Shivering is a response to make the body warmer for whatever reason. And if we are having a fever, if we have our hypothalamus resetting the body temperature, that actually triggers us to shiver so that we can increase our body temperature. So if you're shivering when you have a fever, it's because it's part of the process of getting your body temperature up. Now, what brings it down is vasodilation, sweating, right? These are things that, for instance, if you were exercising and needed to cool down, this is what your body would do. So first we shiver to raise the body temperature. We don't want to stay there too long. So eventually we bring it down by opening the blood vessels, sweating, and then our body temperature falls. This is called the crisis. If you've ever heard that term, then that's the time at which actually the fever breaks, right? So there's some clear advantages to having a fever that you may not know. And that's because, first of all, it increases the levels of transferins. Does anyone remember from our last class what transferins are or why they would be helpful? Do you remember them being associated with iron? They suck up iron, okay? Now, why is that useful? Why do we need iron? Yeah, it's in our electron transport chain, it's in our red blood cells. Why do microbes need iron? Because they also do respiration. So if we're in a war with microbes, one of the resources that we're at war about is iron. Now, microbes have a way to scavenge iron and try to get it away from us. Does anyone remember what they use? It's a really weird word that starts with an S. Siderophores. Do you remember this? So siderophores are what they're using to try to get the iron. We fight back with transferins. I'm sorry to tell you that the microbes will win, but we'll do our best. Okay, so what transferins are really good at is if there's iron in the blood, just free iron, they'll take it so the microbes can't have it. So that's one thing that fever causes is actually increases transferins. It increases the levels of interleukin-1, which further sort of in a positive feedback loop increases the fever because it's talking to the hypothalamus. And also it is an intercellular messenger to promote other things. And also increases the production of interferon, which we'll talk about in a second. Now, there are obviously some disadvantages to having a fever, otherwise we would have them more often and we wouldn't really worry if someone spiked a temperature and we wouldn't take them to the ER and put them in an ice bath, right, if they were at 104 for a couple of days. So one of the things that can happen is it can change the pH of your blood. That's not so good. You can become very dehydrated. Various sort of basic functions of your body can start to fail. And if we hit this temperature, then this will kill us. So there's a pretty narrow window. We don't want to have a fever that's too high for too long. All right, so inflammation, fever. Now another component of our innate immune response is antimicrobial substances. These are things in our blood and lymph that fight pathogens, but again, non-specifically. These are deployed and they're generally helpful, but they're not triggered by specific pathogen. They have widespread effects. And one of the major ones we're going to talk about is the complement system. We'll also talk about interferons, as I mentioned before. Transferins we have already discussed, so I'll just mention them again, try to drill that into your head. And also AMPs, we'll talk about these for a half a microsecond, antimicrobial proteins. So the complement is the most important part of this. And the reason that it's called the complement is because it helps augment the ability of your adaptive immune response, which is, again, the antibodies and the T cells and the B cells that we're going to get to in Chapter 17. It boosts their efficiency. So it acts as a complement to. But as you'll see, it's not specific. The complement is a series of 9 proteins. called C1 through C9. So that's exciting, complement 1 through 9. And they're always in your body. They're just floating around in your bloodstream. They're actually in your tissues all the time. And they're usually inactive. Why is that important? I haven't told you what they do yet, but why would that be a good idea? To have them not do anything. We'll talk about what the outcome is in a second, and that will help you to answer that question for yourself. But basically, they're typically not doing anything, and you actually have to have something happen to turn them on. So there's a couple things. There's this central cascade, right? So these are proteins C3, C5, 6, 7, 8, and 9. This is not all of the 9 proteins. Those are the ones that are considered sort of the core. And what you'll see is that there's a few other ones that work up here in different ways. Of all of these proteins, which ones does it seem to you like would be the most important one? Just based on that diagram. C3, okay? So I want you to remember that. I promise I will ask you this on a test in some way or another. But C3 is considered the central, most important complement protein, and we're going to see why in a second. So there are 3, once you turn the complement on, there's a bunch of ways to do it. We're going to talk about how you activate the complement. But once you activated it, which means basically once you've activated C3, that sets a whole bunch of things in motion. And they always happen. No matter how this gets turned on, then all of these things happen. Okay, so what are those things? One thing is inflammation, which we've just talked about. We've talked about why it is helpful and what it is all about. But the complement, if it's turned on, will set inflammation in motion. Also, there's once you turn the complement on, it basically attacks foreign cells, especially bacterial cells, and what it does is it makes holes in them. So it's like taking a gun and shooting holes in your enemy. It's called cytolysis because you're lysing the cells, and we'll talk about specifically how that happens. And the last thing that this complement system does is something called opsonization. And opsonization is like painting the surface of the microbe you're trying to get rid of with a flag so that your white blood cells know this is a bad guy, get rid of him. So it sort of enhances the ability of white blood cells to find targets and destroy them. Okay, so there's also, as I said, several ways to turn this on. So once you've turned this on, you get all of these outcomes. There's several ways to do that. One's called the classic pathway, one's called the alternative pathway, one's called the lectin pathway, and we'll look at all of those. All right, so I just want to remind you, though, that these 9 proteins in your body are normally not activated. Now that you know what will happen once they are activated, right, that you get inflammation, you start shooting holes in things, and you are flagging things for destruction, why would you want to keep this inactive unless you really needed it? Any idea? Yeah. Yes. It uses energy, and the inflammation process is one of the more important reasons we keep this inactive. Do you want inflammation in your body unless you really need it? Inflammation will bring stuff, to things and set dangerous, destructive cells in motion, but it's not super specific, so you could cause body damage, right? People who have inflammation diseases have problems because their inflammation response is unchecked. So these proteins are always around because we might need them at a moment's notice. You could step on a nail at any time. You could get cut with a knife and then you need them right away. But we have them turned off most of the time. And once they're turned on, they come on really fast and then they go away. Okay, so it's a very fast response, but then it goes away. It's another way to think about it is if we consider that these guys are like first responders to the scene of an emergency, you do not want to be at your house and every 5 minutes have firemen banging your door down just in case there's a fire in your house, or policemen, not just shooting bullets into your backyard. All right, so how do we turn it on? Okay, we activate it in a number of ways. These are the 3 ways. One of them is called the classic pathway, and that is turned on by antibodies binding their antigens. And I want to emphasize that this is something that we're going to talk about in much more detail in Chapter 17, because that's when we talk about adaptive immunity. And what do we know about adaptive immunity? That's where we make antibodies to things, so that's very specific. Okay, so remember I said that these 2 branches of immunity talk to each other. This is one example of that. The specific response of a specific antibody against a specific virus or a specific bacterium or a specific parasite, that will trigger this non-specific complement system to be activated. That's not the only way we can turn the complement on. A lot of the things that turn complement on are accomplished by pattern recognition. So even though they're not responding to a specific bacteria or a specific virus, there are ways your body has of responding to patterns. A lot of bacteria have things on their surface that are unusual that our body cells wouldn't have. And so we have ways of saying, oh, well, I'm not sure what that is, but it's clearly not supposed to be here. We're going to turn the complement on and get rid of it. So one of them is called the alternative pathway. And that one is triggered by these 3 proteins, B, D, and P. They're just other proteins besides the complement that are floating around in your body, and they're pattern recognizers. If they recognize a pattern that is common to bacteria, they will just glom onto it, and when they bind the bacteria, it causes a chain reaction that activates the complement. The 3rd way is through the lectin system, and lectins are just proteins that are very specific to binding mannose, which is a sugar, and that sugar is found very commonly on the surface of bacteria. So if lectins are binding mannose, that's a signal to our body that something is wrong. Now, we've already talked about this, but there are, whichever way you turn on complement, you get all of these same effects. So once you turn it on, you have opsonization, which is just painting the microbes that you're trying to get rid of in order to make them more visible to your immune system. We have inflammation, so we're bringing in more white blood cells and other first responders to the scene. And we have cytolysis. So there are a variety of proteins we haven't really talked about yet that actually punch holes in the membrane and make it easier to destroy the microbes you're trying to get rid of. So here is, again, the core cascade. There's 9 complement proteins. We're going to talk about a couple of more of them than this, but these are the core. And the most important of them, of course, C3, because he's at the top. So the first step in activating the core cascade, no matter how you set complement in motion, the first thing that happens in all of those cases is that C3, which is the inactive component, gets chopped in half. And when it's chopped in half, both pieces are now active. They do something different than they did when they were stuck together. So one of those pieces, called C3a, goes and binds to mast cells, which are immune cells that release histamine. And you'll remember that histamine is this chemical that is frequently associated with allergies, which is why we take antihistamines. Histamine is the main chemical that your body sends out to start the inflammation response. And that's why when you have allergies, you're having an inflammation that you just wish you weren't, right? So half of the C3 protein helps to initiate this histamine inflammation program. The other half of C3 talks to this other protein, C5, chops it in half, and these 2 pieces, which were, when they were stuck together, not active, as individuals they can do something. So this half of C5 goes and binds to mast cells, and together, these 2 pieces trigger inflammation, increase blood vessel permeability, vasodilate, and send out a lot of signals to bring in phagocytes to the scene. The other half of C5 talks to C6, which recruits C7, 8, and 9, and this entire thing forms a hole, and it's a big pore, but basically you want to think about it as like a bullet or some way to punch a hole in a microbe. So they go and find a microbe and they make these holes. And then what happens? You have holes in the membrane of your microbe, usually bacteria. Why is that bad for it? All the stuff inside is going to leak out, right? This is bad for all cells. And you'll notice this is a recurrent theme. This is one way that, using different proteins, but microbes will try to kill us by making holes in our membranes. We try to kill them by making holes in their membranes. But this is the part of complement that is called cytolysis, because you're making holes in membranes and the cytoplasm is leaking out. And that's clearly fatal for cells. The last thing that I haven't mentioned is that this half of C3 also is part of something called opsonization that I'm going to cover in more detail, but it's part of this painting the microbes so that it makes it easier for macrophages and other phagocytes to find them. So, if C3 is the most important and cutting it in half triggers all of these effects, how do we trigger C3? What is happening? So as I said, there's something called the classical pathway, which is one of the ways to turn on the complement system. And what happens is if there's a microbe and it's got something on the surface that we have already formed antibodies to, okay, so this is again part of that specific immune response. We have antibodies against this very particular pathogen. The antibodies bind to the surface, and antibodies have this Y shape. The two branches of the Y are what grab onto the antigen that they're specific for, and this stem here, when it's sticking out like this, is a site where this protein C1 that we haven't talked about yet likes to bind. Now, when this is bound to antibodies that are bound to their antigen on some pathogen, then this causes C2 and C4 to become activated, by which I mean they get chopped in half, and these previously inactive proteins, these two pieces are now active. And what primarily happens is that this half of C2, C2A, and this half of C4, C4B, get together, and they activate C3, right? And then what happens? If we activate C3, it triggers all kinds of things. There are three things in particular. When we set complement in motion, what are the three outcomes? Remember what C3 gets chopped in half and it goes off to do various things with the rest of the complement system, which includes what? Inflammation, making holes in things, or cytolysis, and flagging things for destruction, opsonization. Okay, so that's one way to turn on C3. This is the alternative pathway. So as you might imagine, historically, this is what we learned about first, so we called it the classical pathway. Then we found this one and we went, well, we're going to call it the alternative pathway because it's not doing the same thing. But this is what I told you. Here's a microbe, and there are specific fat-sugar combinations on bacteria that are unusual, that are not found on our body cells. These three proteins, B, D, and P, just run around in our bloodstream looking for those combinations. They're not going to bind to our cells because we don't have those combinations. They're just pattern recognition molecules. And when they find a target, then that triggers C3 to bind to them and become activated. So it gets chopped in half, and then what happens? What are the three outcomes of the complement? We get inflammation, right? So calling vasodilation, sending out chemical signals, first responders, and especially white blood cells come to the scene. We start punching holes in the membranes of this microbe. We have opsonization, so we're coating the microbe in order to make it visible. Last but not least, we have the lectin pathway. And again, this is another pattern recognition one. So it happens a little bit later sometimes in the infection, and primarily happens because some phagocytes release, basically if they take in a pathogen, it triggers them to release chemical signals. Those signals go to the liver. The liver makes extra lectins, and lectins are proteins that really like to bind mannose, this sugar. And so they'll go out roving around looking for, hey, microbes that have mannose on them. binding lectin to this mannose on the microbe surface activates C2 and C4, which get together and activate C3. And once you've turned on C3, what happens? Inflammation, opsonization, cytolysis. Okay, so we're just going to take another quick look at these three outcomes, although we've talked about them quite a bit. So we're going to look at inflammation, we're going to look at opsonization and what exactly is happening, and as well we're going to talk about cytolysis, or what I like to call the MAC attack, the membrane attack complex. Okay, so inflammation. Here is, remember that after you chop C3 in half, one of the things that happens is it activates C5, and half of C5 binds to mast cells, half of C3 binds to mast cells, and this together is like a trigger to release histamines, and you need both of these, okay? And if you want to think about it this way, and it's a little bit of a dark illusion, but remember that, you know, if we have nuclear weapons, a lot of times people have multiple keys. You have to have both keys or 3 keys, and they all have to be inserted, and so there are a lot of fail-safes if you have a really dangerous weapon. So inflammation is a somewhat dangerous weapon. You need both of these things in order to turn this on, so you don't get it by accident. Once histamine is released, it triggers all kinds of things associated with inflammation that we've talked about, including vasodilation, including bringing more white blood cells to the scene. So that's basically how inflammation works. Now, opsonization is, we're going to actually talk about this in a couple of ways. So in this part of the course, we're going to talk about the contribution of C3. So when you activate C3, again, it gets chopped in half, and half of it is directly able to bind to the surface of bacteria and other microbes. And by doing that, it's like coating it with almost like with a red paint, because there are receptors on the surface of phagocytes that are specific for this C3 protein. So it makes it much easier for these guys to find their targets. It's not the only way. Okay, phagocytes have a number of different receptors that allow them to detect microbes. They're able to do it if the microbe is covered in antibodies, then being covered in antibodies helps them to find that microbe, but also being covered in C3b is very helpful. So once you've coated it with one of these chemicals that makes it easier to find, then the macrophages are highly stimulated to engulf it. And this is helpful because what we don't want is macrophages to be roaming around in your body and just, you know, accidentally eating body cells, right? So we want it to be that there's a little bit discriminated and they don't just eat whatever, right? They sort of have to be stimulated. And the third thing that we've talked about, cytolysis, is a result of the membrane attack complex, or the MAC attack. And what it causes, so here's an example of this is an electron micrograph, a scanning electron micrograph of a bacterium. And this is what it looks like after it's been attacked with these pores and had all its holes poked in it. So again, there are a variety of complement proteins that are initiated by breaking C5 in half. C5 recruits all of these other complement proteins, 6, 7, 8, and 9, and they just form a big hole, and they just poke holes in microbes and kill them. So, do you think, given that this is the way our body responds non-specifically to invasion, do you think that microbes have evolved ways of fighting back? They absolutely have. We are always in an arms race with them, and they come up with something, and we come up with something, but they're better, really, because that's why it's so helpful for us to have an adaptive immune response. That's the sort of ace in the hole, the way that we sort of keep an edge against them. But they're evolving much faster, right? They're, in some cases, their generation time is 20 minutes. So they're going through lots and lots of change much faster than we can. I've talked about this before, but capsules, if you put a capsule on yourself as a bacterium, it can make you more effective as a pathogen. And that's because one of the things that capsules do is prevent complement activation. So remember I told you that being encapsulated, like for instance the pneumonia bacterium, makes you sort of invisible to the immune system. This is why. Because there are, part of capsules is something called sialic acid that is like a coating that prevents that MAC attack, those proteins that form a pore. So it's kind of like having body armor, right? So those pores try to make a hole in you, they can't. Also, that sialic acid prevents opsonization, so you can't be coated. Okay, so now you have like body armor plus a paint-proof raincoat. And in addition, this sialic acid is also found in some gram-negative cell walls. Okay, so capsules and some gram-negatives have this protection against both opsonization and cytolysis. There are other things, particular complexes of lipids and carbohydrates. So some parts of the, again, the gram-negative surface, that extra outer plasma membrane that has sugar attached, and some of them are also repellent to this, to these pores being formed. And salmonella is one example. The structure of this sugar that's attached to the outside of salmonella prevents the pore from reaching that microbe and making holes in it. Another thing that microbes do sometimes is they secrete enzymes that target the complement. In particular, some gram-positive cocci that are pathogens have an enzyme that can digest C5a and prevent it from working, so that you sort of cut one of the legs off of the tripod of the complement system. And I think this is really especially sneaky. There are some viruses that actually, it's almost like a in-your-face kind of thing. These viruses will actually attach to receptors of the complement on body cells to start their life cycle. So they use the very tools that are designed to protect you in order to get into your body. Okay, so that's all I am going to say about the complement. If you are struggling with this at all or feel like that wasn't super solid, there's a mini lecture on this online in the supplementary module for chapter 16 and 17 that goes into this again. And I think it's very helpful because it's kind of a bigger picture overview, and it's kind of touches on what's the most important things to remember about that. So that's the complement. Another thing that we have going for us is interferons, and there are 3 kinds, so we're going to talk about them. They're different, and I'm sorry, you have to remember them. Okay, so there are 3 kinds. There's alpha, there's beta, and there's gamma. And alpha and beta work together. They do essentially the same thing, okay? They're produced by body cells under attack. And specifically, they're produced when body cells have a viral infection. So if your nasal epithelium has a cold, or your, some other part of your body cells are being attacked by viruses, one of their defenses is to secrete interferon alpha and beta. Now, it's kind of, at that point, too late for the cell that's infected. It's more of a distress call that is warning neighbors. So this is more of a neighborhood watch kind of thing. It's not going to really help the infected cell, but it does help their neighbors. And so it's kind of an altruistic thing. What happens is that if you are infected with a virus, you are triggered to make interferon alpha and beta that then you secrete just into the local area, just to your neighbors, just to say, hey, I'm under attack. Those neighbors can't do anything about it. They can't help you. It doesn't really cause white blood cells to come and rescue you. But what it does is it causes the neighbors to initiate an antiviral defense mechanism. They produce proteins that are helpful generally against viral infection. And we'll talk about this probably not any further, but the other kind of interferon called gamma interferon is produced by lymphocytes, and it is part of the way that you activate phagocytes to do what they do. So just remember that alpha and beta are the kind that are produced by body cells when they're under viral attack, and that gamma interferon is actually different. I'm going to now just talk about alpha and beta interferon effects in this just a little bit more to show you in this diagram how it works. Okay, so here's a cell, and it gets a virus. And in the process of the viral infection, the viral genome causes the production of more viral particles. And just the process of your cell being hijacked by a virus triggers the production of interferon. Okay, so there's some kind of communication system that I'm not going to make you know about, but just the presence of this viral infection triggers the transcription of interferon messenger RNA, interferon alpha and beta. They're proteins. They get translated into these proteins. Those proteins, interferon alpha and beta, are secreted. They go next door. They ring the doorbell of your neighbors. And before these guys are exposed to this virus, which is coming, right, because we've got viral particles and they're about to come and target the neighbor cells, they've already received this secret message, and that causes them to make antiviral proteins to protect them from this viral infection and stop the virus from replicating. I've already mentioned this, but I'm going to mention it again. Another component of innate immunity is transferrins. Remember, that is a chemical we make to bind iron in the serum and prevent microbes from getting it, sort of hoping to starve them. They, of course, fight back, secreting something called siderophores, which steal the iron back. And where do they get more iron? Well, some of them have the ability to explode red blood cells, which have lots of iron. So we have this sort of back and forth. The pathogens usually win, but at least we're doing something about it. Last but not least, we've got antimicrobial peptides. These are different from other antimicrobial substances because peptides are small. Peptide is a word we use when we're talking about a protein that's like less than 50 amino acids. Most proteins are at least 100. Some of them are 1,000. So these little tiny proteins, they're really short, are just around, and what they do is they lyse, explode bacterial cells. There's hundreds of them that we've discovered so far, and they're active against viruses, bacteria, fungi, parasites, and that's really all you need to know about them. Okay, so that's chapter 16.